Okay, we're going to go ahead and begin. Thank you everyone for coming. I appreciate your being here, and I know it has been a crazy year. I'm so proud of all of you for actually finishing your projects. It hasn't been an easy one. I can't wait for everyone to tell each other about their projects. And as is typical for the pandemic, we have to have a flexibility band on our wrists to say, okay, we're going to go to plan B. Plan B is that we do not have breakout rooms, and therefore we are going to meet as one group, and each group is going to have only 10 minutes to present one time. For those who are not presenting, what I'd like you to do is raise your hand if you have a question. I won't be able to monitor both the chat and the participants very easily. So, but I guess I can do that. So you can either type your questions or you can raise your hand and I'll recognize you to ask a question. So what I'd like teams to do is share their screen if they're going to use their some kind of an electronic version, or if you're going to use your model, you want to hold your model up to your camera so that we can see it. And you may be going back and forth. So if you're sharing your screen, you go down to the bottom of your Zoom meeting and you'll see something green that says share screen. And then when you're done, if you will unshare your screen, there will be a little red tag at the top that says something like turn off sharing so that the next person can go. And I will be the monitor of the questions, and I will be the monitor of the time. So the first group, and I will just give you an idea. We're going to have the Grandview drug design team start first. And then we're going to have our three groups that did RDRPs, the RNA Dependent Rna Polymerase. So we'll do MSOE, then Lane, then Grandview. And then we'll switch to the spike protein and do Hamlin and MSOE and Nova. And then we'll conclude with ORF8 with Campbell. You don't have to remember that. I'll tell you who's up next, but if you at least got an idea of where you are in the stream, that is fine. Okay, so Jesse, are you ready to go ahead and do the Grandview drug design team work?
Oh, I'm sorry, I was muted.
I'm mute too.
Yes, I'm ready. Can everyone hear me? Okay, I lost audio. One moment. Okay, so I'm here. My name is Jesse. I'm here representing Grandview University, and my group is interested in the SARS-CoV-2. So we spent the last two semesters studying SARS-CoV-2 or drug design in the context of one of its proteins called the main protease, or NSP5, that's involved in viral replication. So we began with a paper that had identified a lead compound called N3 as a potential inhibitor of NSP5, and we began modeling our own drugs in silico to bind the N3 active site. We concluded our project by making a physical model of one of our better compounds, as well as the active site and one of the entire NSP5 protein, and the latter two are available on our poster. Thank you.
Great. Do you have your model with you or not? Oh, you're muted. Don't mute while you're presenting.
Oh, sorry, I don't have the models on me now.
Okay. Did you want to show us some of the images from your JMOL exploration?
Yeah, I have to pull them up one moment here. Sorry about that. It actually might take me a moment to access them. I'm sorry. Is that all right?
Say what?
It actually might take me a moment to access them. I'm sorry. I had them on my other computer.
Oh, okay.
I can have them for the breakout room, though. I apologize.
So while you're working on that, and I don't know if you can do both things at the same time, I saw that you designed some drugs and docked them.
Mm-hmm.
What docking software did you use?
We used PyRx, PyMol, JMol, and I believe that's it.
Okay, did any other groups, and this is where if you did, you can unmute and jump in. Did any other groups do any docking this year? Oh, somebody's raised their hand. Let me look. Oh, nope. We normally use Vina. I don't know what other people have used. So I'd be really interested in knowing more about how you did that. And you also said you used some certain rules, a Lipinski's rule of 5, Haber's rules. What are those rules?
So they have to do with the surface area, the polarity, and something called the log P values, which we played around with. And oh, I'm sorry, we did use one other, Marvin sketch, which was how we modeled some of our compounds. So we played around with those, looked at their properties in Marvin sketch, and ran them back through PyMol and PyRx to get docking information.
Okay, and we did have a question asking, what did you use the Marvin sketch for?
Marvin sketch was how we designed other compounds. So we would take some other inhibitors that were in the paper, like synincerin, epsilon, and we modeled our compounds off of those, but we changed some of the properties to affect polarity or the surface area or something like that to see if we could get a better delta G value for our binding, for our docking study.
And then the other question I have, Dr. Schiffer, you have a question, and I hope you're going to ask the one that I'm thinking of.
Oh, I don't know what, you should go ahead and ask what you're thinking of.
Well, I'm hogging it, and there are other people who have their hands up. So I'm going to stop while we get everybody's hands, and then.
All right. Hi, Jesse. My name is Celia Schiffer. I am a professor of biochemistry at University of Massachusetts Medical School, and we're actually studying MPRO in my lab right now. We've solved a bunch of crystal structures of it and are doing docking. So it's really cool that you chose this as a project. I was curious, what struck you about MPRO that was the most fascinating? Why is it such a good drug target?
The main property of MPRO that we were looking at was that it's highly conserved among variants of COVID, so it's a good target for drugs to last a long time.
Yeah, no, that's definitely true. The other thing that's kind of cool about MPRO is that it has to cut 11 different sites in the viral polyprotein, which is a lot of different sequences for one protease to recognize. And what other thing about MPRO is it a good target for antiviral?
Well, an effective inhibitor would essentially stop the drug from replicating, so that would be a really effective drug target.
Yeah, and do you remember what kind of protease it is?
I forget off the top of my head. What exactly are you looking for?
It's a cysteine protease, so you can actually have covalent inhibitors, which is.
Well, yeah.
No. Well, thank you. I'm glad you're working on that important drug target.
Thank you.
Okay, Amber.
Yeah, hello. So I think when I was looking at your poster, was there like 3 or so kind of molecules that you tested for docking? And then like, I guess, how did you choose those molecules as for the
I'm
ones
sorry,
that you
you're
were
really quiet. I can't quite hear you.
Can you hear me better now?
Yeah, that's better.
Okay. I guess like I think I saw something about you testing like multiple molecules to see how they docked, right? And so I guess my question is, how did you decide that those were the molecules that you were going to test?
Well, we played around with some of the Lipinski's rules, like the surface area and the polarity. So we came up with a bunch of different types of the changes and just ran them through and just ran a bunch of them through to see what the docking study came out as. And some of the things that gave us better binding affinities were larger surface areas. One thing I did was I changed all the polarity on one side and then all the polarity on the other side to the opposite charges, and that seemed to have a positive effect. But you know, those were just some of the things we played around with, and we happen to have a couple that turned out to be better than the ones that we started with, and those involved larger surface area for one and just some polarity changes.
Bergen?
So I was looking at the structures of the.
Oh, Bergen, we lost your voice.
Is this better?
Yes, you got it.
Okay. I was looking at the 3 molecules that the drug molecules under JMOL exploration, and earlier you mentioned the log P values, which I'm not super familiar with drug discovery, but I'm certain I think I remember them being like the a ratio of their solubility in oxygen and water, so like how polar they are. I noticed 2 of the molecules seem to be fairly hydrophobic and aromatic, whereas the N3 one would seem to be a fair bit more polar. Are you using those different polarity of molecules just to test like how polarity affects their binding, or is there different rationale between why you test these molecules?
Right, N3 is a really large molecule. The ones we were testing are much smaller.
Okay.
So some of it was just playing around and seeing what happens, but some of it was just increasing polarity of the molecule, and some of it was increasing polarity in specific places. Like I said, that one of them I made more positive on one side, one negative on the other side. But a lot of it was just trying a lot of things and seeing what stuck.
Okay, we've got one more. Dr. Sikora, if you'd like to ask your question.
Yes, I was wondering about the actual affinity of your drugs. What kind of delta G's did PyRX give you?
The better ones were around -7 kilojoules per mole.
Okay.
So I think the one we started with, I think cyninsarin was one of the compounds that was the one. Epsilon and cyninsarin were 2 of the smaller molecules that were modeled on the N3 active site. Those were giving us around 6, I believe. I forget the exact number, but around 6, and we got up to about 7 with some changes.
Okay, Dr. Guzman, we'll take your question and then we'll move on.
So Jesse, it's Karen Guzman from Campbell University.
Hi.
I was just wondering, can you tell us more about what the protease actually recognizes? And if you know anything about it, you know, Dr. Schiffer mentioned that it recognizes 11 different sites. So how is it recognizing so many? Is it that all those sites have some common feature that you can tell us?
Ooh, that's a good question. I believe that there's a cysteine involved in the active site for N3, but as far as what it's actually recognizing, I'm not completely sure. I remember there was about 11 amino acids that we were targeting. The protein, where the protein is binding, I'm not completely positive what it's recognizing. So I would have to look into that.
Thanks.
Okay, we're going to have to move on to the next one, and the next one is the MSOE RNA Dependent Rna Polymerase. We're going to do 3 that are about RNA Dependent Rna Polymerase. So go ahead.
Okay, hi, I'm Muskan. I'm here with Amber and Natalie. So this time we chose to explore, obviously, like Margaret said, RNA-dependent RNA polymerase of SARS-CoV-2. That can also be called RDRP. And this is basically an enzyme that catalyzes RNA synthesis, and therefore it helps you like replicate the virus's genome. Remdesivir is, you've obviously heard about it. It works to inhibit the RDRP because it's a nucleoside analog. So it basically resembles adenosine, so like your A base, really closely. And this basically causes RDRP to recognize it and incorporate it into like your newly synthesized RNA strand. And then it actually terminates RNA synthesis about 3 base pairs down from where that remdesivir was incorporated. So that's a little about our model. Thanks for listening, and let us know if you have questions.
Yeah, and I'm going to share my screen to our e-poster so we can show off a little bit of the model real quick.
Okay, do you have your model with you?
I do have it with me, but I think it's easier to see on JMOL, but if you want, I can show off both.
Well, I looked at the image on your e-poster, and I really wanted to see on the model where the thumb and the fingers and the palm of your, I know a lot of RNA polymerases are described that way, and I would love to see that in the model itself. But if you want to show what you're going to show, that's fine.
Sure. I think first off, I guess I know I didn't look too closely into exactly how these structures that are being pointed out like lie directly on our model. I mentioned like in the e-poster that some of these structures are actually missing in the PDB file that we used. And this is a more recent kind of structure that was kind of found. And so it's a little bit more updated, but it shows you a little bit more on how like the structure kind of like helps to stabilize the RNA. And as you said, like kind of those, I guess, structures that are often more recognized, so like the palm and the fingers in different domains here. I think I was going to show off kind of at least this JMOL script. This one shows off where the remdesivir is bound in our PDB file. And you can see how it very much blends in with the RNA bases around it. It looks just like it without the extra coloring. It'd be pretty hard to tell that it's very much different, which is how remdesivir works, as Muskan said. And if you look at like, this is adenosine, and it'll swap over to remdesivir next, but you can see that there's a lot of similarities, along with a few differences, which are what is theorized to like cause the termination in the RNA synthesis. I think the biggest differences is there's this cyano group on the back here that's not part of adenosine, and then also there's like a carbon-carbon bond here instead of there being like a carbon-nitrogen bond in adenosine. So yeah, that is our model, and I guess stop sharing.
Do we have any questions? Oh, you're going to show us your model. Show us.
Say, you can't see it as easily, which is why I wanted to show it on the JMOL exploration, but you can kind of see the structures here as you saw it online. So yeah, we can take questions now.
Bergen.
So you said remdesivir is a nucleoside analog, correct?
Correct.
So it's a little unrelated of a question, but it's my understanding that like in some chemotherapies, they use like nucleotide analogs that kind of get inserted into human DNA and cause a cell to generate mutations that cause apoptosis. Is using a nucleoside analog in a treatment like this have like the potential for screwing with the genetic functions of the human cell, where those nucleoside analogs could maybe mess up like mRNA transcription or the expression of other proteins?
I don't think so. I guess in the sense of like the nucleoside analog, the remdesivir, isn't going to be in the nucleus, right? And so it shouldn't interfere with RNA being transcripted made there. So I don't think it would mess with it, which is probably why it's a viable drug.
Okay, I mean, obviously it's a viable drug, so it wouldn't cause cancer, but I was just curious about the mechanism behind it. Thank you.
Do you have any idea? I see the other hand came up when I was looking down. Do you have any idea why remdesivir works for SARS, but as it turns out, doesn't work so well for SARS-CoV-2?
I'm not 100% sure of why it worked for like MERS. Though I think when I was looking into it, I think they found that maybe it also wasn't a super effective drug there, but I don't remember too well if that's what I was finding. And so I don't fully know why they thought that, like, because in theory, like it, in terms of like how it's working, it is disrupting the RDRP. But I don't know if it was like the environmental conditions that just made it to where it wasn't maybe getting uptaken as much, or it just wasn't working. So I'm not 100% sure why it doesn't work, but maybe one of the other teams found something that will speak more to that.
We can ask that question as well.
I just was curious, how many different macromolecular entities form the full complex?
Yeah, so I guess I can bring up the model quick since you can kind of see it on here. But as you can see, there are these three like large different colored like portions on our model, and that's because they each represent a different non-structural protein. And so to my understanding, there are three major non-structural proteins that come together and form a complex that allow the RDRP to function the way it does. And I think it's NSP7, NSP8, and NSP12. Though I think the newer model that I was looking at that was on the e-poster, it said that there might even be two NSP8s on there. But that could definitely be something to look more into as more research is done on that.
Thank you.
Birkin?
And your answer to the last question, you mentioned the non-structural proteins, so NSP 7, 8, and 12. I'm just curious about the semantic aspects of like the non-structural protein. Like, can you elaborate like what that definition means? Because with how important protein structure is to function, why do we call them non-structural if they confer some structure? What's the meaning behind that?
To my understanding, like the non-structural proteins aren't involved in like the kind of, I guess, what you might recognize as COVID, right? So like you've got your membrane protein, your spike proteins. I think like maybe like, I think there's 4 structural proteins. You've got your spike, your membrane envelope, and there's something else in there too. I can't remember.
So non-structural relative to the virus, not the function of the protein itself.
Yes, and so non-structural proteins have other functions, right? They do things, but they aren't like actually a part of the structure of like the kind of outer coating and whatnot and what's holding everything together.
Okay, thank you.
Dr. Guzman?
Amber, I have a question. You may not be able to answer it, so maybe somebody else can help us with it, though. It's basically, I may be the only one, but all of these different ORFs and structural proteins get me confused. And the fact also that they get multiple names. So in some cases, I've heard NSP8 is actually also ORF8, which we studied, but I can't imagine ORF8 as being part of that that structure that you're showing. Does anybody know if NSP8 is ORF8 or if sometimes it refers to another protein? And like I say, you may not know. I'm kind of opening this up to everybody.
I think open reading frame 8 is in the 3' end of the protein of the genome, and the non-structural proteins are in the 5' end of the protein. Mm-hmm. And all of the non-structural proteins are the ones that are involved in replication, transcription, whatever it is, turning off the host immune system. And in the 5' end, or the 3' end, that codes for things that are the structural proteins like the spike protein. And they vary a little bit in different coronaviruses. And so until we know what those things are called, or what their functions are, we call them open reading frames, which is why they're ORF, and they're numbered in order. Some of the proteins are named and some of them are numbered. And the numbered ones mean when they were numbered, we didn't know what they did.
Okay, so our ORF8 is not NSP8.
No,
that you saw. Okay,
your
good.
ORF8 is in the 3' end of the genome, and NSP8 is smack dab in the middle or a little bit. So there are like 15 NSP proteins that are made from the 5' end.
That helps.
Okay. Thank you, MSOE RDRP team. We've got 2 teams there, so I'm gonna thank you for that. And we're going to move on to the Lane team. And they also did RDRP.
Hey, I'm Michael Davis with the Lane College team.
Yes.
I have both. I want to show both my physical model and a tutorial while I speak. So we chose to do ours on the RDRP complex with the remdesivir. Remdesivir is the only FDA-approved antiviral drug for COVID-19 targeting enzymes, RDRP, but only for severe cases as it causes chain termination. Let this load while I talk. So here, I also show this as well while I play, just in case it moves faster or slow. We highlighted the non-structural proteins, NSP7, which we have highlighted in green, and NSP8, which we have highlighted in red, and NSP12, which we have highlighted in blue. And then you can also see the active site with the RNAs as well in the middle with the yellow, which is the remdesivir, which is highlighted right now on the screen. And then we also have the RNA primer, which is pink, and then the RNA template, which is orange. And then and as they had stated before, the AMP and the remdesivir, they're very similar. And she had stated that remdesivir has a cyano group and a carbon-carbon bond instead of a carbon-nitrogen bond. And though these differences, it may seem, it may not seem that much to the eye, but this causes the remdesivir to be unstable, making it more likely to lead to chain termination. And I also show that in these models as well. And you can see that as well with our RNA template and our RNA primer. As the RNA template, it keeps going, but however, you can see where the active site would be. The RNA primer stops and the chain termination is here, where the active site would be. That's it. If I have any questions.
Were you going to show us your model as well?
Oh, yeah.
So you'll have to unshare your screen.
Oh.
We can't see you, we just see your screen.
Right, I'm looking at the, let me stop. So as I was talking, I was showing this, I had forgot about that. But I was saying, I was highlighting we have the NSP7, which is in green, the NSP8, which is in red, and the NSP12, which is in blue. And then you can also, well, you can kind of see the active site. I hope you can see it better on the JMOL tutorial. But you can see the adenine and the remdesivir bound in there closer. The remdesivir we have highlighted in yellow. And then, like I was saying about the RNA template and the RNA primer. But then we have other models to highlight this better with our remdesivir. and the AMP. So like I was saying before, they're very, very similar. It was kind of hard for me to find the differences at first when we first began our project, but the remdesivir has a cyano group and a carbon-carbon bond right here, which we have in yellow. But on our AMP, it doesn't have the cyano group, and then it has a carbon-nitrogen bond. And that's why I was saying it didn't look like when I first was doing the project, it was hard for me to spot the differences, but to understand. And when I first seen it, I was like, oh, there's not much, but these differences, these are important because these are what makes the remdesivir less stable, which leads to the chain termination.
I really like you use you built those small molecules because that makes it much easier to see.
Right.
Schiffer?
Thanks very much, Michael. That was a really nice presentation. I was curious if you've thought about how the RNA gets into the middle of this complex and what conformational changes must need to occur for that to happen, and then the remdesivir to get into the active site.
No, I have not.
Okay. That's okay. That's okay. But these proteins are, the crystal structures are just a rigid snapshot. So in order for them to come together, there has to be a lot of changes and dynamics involved. So we get snapshots of the picture of how the complexes form, but how it actually comes together means that there's a lot of flexibility. So just something to think about. Thank you.
Okay, Dean.
Thank you. Michael, I should say first, I thought that's a really nice way of presenting it with the poster, and it was neat to see your model as well. Curious, when you say that it's less stable, are you saying that it doesn't stay in the active site for as long, or that it prevents the active site, it sounds like, from doing its job correctly? Can you elaborate a little more on that?
I guess I'm saying that I really.
Sorry if I threw you a curveball there.
Right. I was like, I can't answer that question. Dr. Vanshar, could you help me?
Well, I was going to ask if any of the other teams that studied RNA dependent RNA polymerase have an answer for that. If you do, could you raise your hand? Amber?
Yeah, it's not too much of an answer, so I don't know how much it'll, actually expand. I remember when I was looking at papers, I think that there's a lot of kind of theory going on for exactly how it destabilizes the kind of RNA chain. But a lot of people think that it has to do with the cyano group on the back and how it's interacting with the backbone of the rest of the RNA. And that's somehow causing determination. So hopefully that helps a little bit.
Thank you.
And does anyone know, does the RNA dependent RNA polymerase in SARS-CoV-2 have a proofreading function? Amber? And then we'll take Karshana.
I don't know for a fact, but they very rarely do, right? Proofreading functions on something that has such an error-prone nature.
Yeah, I seem to think that this one was a little different, but Karshana, do you know?
Yeah, actually, the reason as to why MERS and SARS-CoV-2 have such a difference and why remdesivir works is because that SARS-CoV-2, unlike most viruses, actually does have a proofreading protein, and it cuts it it cuts it out of any so although it mimics adenosine, it cuts out remdesivir because it knows that that's not the actual nucleotide that needs to be there, and it will replace it with actual adenosine. So therefore, that's why it doesn't work as effectively as it does in mares, because that didn't have as strong or didn't have a proofreading protein.
Okay. Thank you. I also thought that I had read that it had a proofreading function, which was one of the reasons I think we didn't get variants earlier on in the pandemic, but we do seem to be collecting variants now. So it might proofread, but it might not be a perfect proofreading, but it's not taking the remdesivir very well. So Michael, thank you for that presentation. And Karshana, are you the next person up? Okay, we're going to let you go ahead and talk about your protein.
Of course. Thank you for having me here today. Well, I'm Karshana Kalyanaraman, and I am part of the Grandview University RDRP team from Des Moines, Iowa. And as the two other teams had already discussed, we also focused on the RNA Dependent Rna Polymerase, because we found that that was a really important or really significant or kind of eye-catching when we first picked the topic. And we wanted to focus on how remdesivir was supposedly how it would have inhibited the further chain elongation of the viral RNA. Here, I can show you here that there remdesivir is inserted as a prodrug and then loses this part, which carries it in and becomes phosphorylated with 3 phosphates here, and it mimics adenosine, as they had pointed out earlier, and therefore it goes and it binds onto the RNA. Here you can see in my model, and I'll show you here that remdesivir is right here, and it there's these hydrogen bonds highlighted in yellow, and that is where it binds on, and is supposed to inhibit elongation, but that proofreader protein would have cut that out, and the primer strand would have continued to replicate, but that was the hope for it to be like that. Here you can see in our poster that that's where it was highlighted, and those yellow bonds are the hydrogen bonds. But we also found that there were, within the active site of the entire complex, there were the active site where the RNA went through, and remdesivir had some side chain interactions, and you can see that it's blinking right now. So the one that's blue and flashing, that one is the tyrosine 687, and then the other protein is the lysine 545 that has it here. You can kind of see it. It's very small. It's very small, but you can see it right here, and then you can see it on the other side. And those are the active side chain interactions that we wanted to highlight in both of our models, and show how remdesivir was supposed to be a potential therapeutic drug.
Okay. Are there any questions? Thank you. Amber.
Yeah, so I guess when I was reading about the exonuclease, I think I found some papers that were talking about how it seemed that remdesivir was maybe somewhat more resistant to kind of being recognized by it. And so I guess I wanted to ask about what it does seem to be recognizing, because I didn't look into that quite as much as it seemed that you did. So I guess I wanted to know what your understanding of that was.
I recently was pretty curious about that too, and I looked at a study that Stanford had done about why SARS-CoV-2 had not worked as effectively, and it was due to that proofreader protein. And it did recognize it, and as that cyanide group is, the C triple bonded N is really important, or kind of standing out compared to adenosine. That's why it was able to see chemically that it wasn't the same as adenosine. It was able to recognize that and more effectively pull it out than what usually viruses can do.
All right, thank you.
So I was really curious. In your poster, you showed that remdesivir comes in as a prodrug. And I wondered, that was the first time I knew it came in as a prodrug. Do you know why they don't just give you remdesivir, the active drug?
I don't really know as to why they do that. I think it's probably because of how it's like given in, or like transferred into the body, or it might not go to where it needs to go in the where it should be. That's why it's delivered through that, and then it becomes phosphorylated. So I should look more into that.
Okay. Cristo, you've got a question?
Yeah, thank you. I was wondering, how could remdesivir be modified possibly to like increase its efficacy in preventing like further RNA synthesis and like viral replication in general for SARS-CoV-2?
That's a good question. I don't, because it's a proofreading protein, that seems a little bit more effective in taking out remdesivir. I did not, we didn't really do much drug design as the other Grandview team in seeing how we could improve the efficacy, but I think that's a really good future direction for this.
Thank you.
I saw another hand. Did that come down? Dr. Schiffer and then Berrigan.
I just was going to ask the same question I asked Michael about the dynamics. If you thought about how the dynamics of this complex enzyme works.
I'm in the same boat as Michael. We didn't really look into the dynamics of that, but I think that would be really interesting to see how it works.
Thank you.
Berrigan?
This is kind of piggybacking off your question about the prodrug structure. I noticed that in the prodrug structure it doesn't have a charge, but when it's in its active form, it does. Is it possible that the active drug, like when glucose is phosphorylated, it can't cross membranes anymore? Is that a likely reason as to why it's administered in its prodrug form, so it can cross cell membranes? I'm not sure if the drug actually works inside the cytoplasm or in a different membrane. Could you elaborate?
I think it's just due to being phosphor. I don't know exactly how that prodrug is different, or like does it cross through membranes through that, but that could be a definite possibility.
Okay, Dr. Martinez.
Hi, I wanted to go ahead and read a comment that Dr. Schiffer put on the chat, since I'm helping monitor the chat. And she's clarifying that NSP14 is the exonuclease, so exo-N domain that proofreads the nascent RNA strand and excises the misincorporated nucleotides. So to add to the proofreading conversation.
That's great. Thank you. Before we go on, I wanted to make a general comment about these three posters because they covered the same topic, but you all approached it a little bit different way. If you haven't looked at the Lane poster, I really liked it because in addition to having the JMOL rotating, they had the plain images and just snapshots so you could compare them. And I think that the Grandview team also had some structural things there. It's kind of hard to compare the structure of two things when they're rotating in space. or when you're seeing one and then seeing the other. So I liked having those images there. That helped me to make the comparison. And the other thing that was really hard with this protein is the active site was buried so far inside the structure that it helped to have the RNA out or the small molecules so they could be compared. So you should consider when you look at other teams' projects, you were telling the same story, but you found very different ways to tell them. And I thought that was great. Thank you to all three teams for doing that. And now we're going to switch over to the spike protein. And we're going to start with the Hamlin team's spike protein project.
Thank you. I'm Dean.
I'm Lauren. I'm Trisha.
And we started out our project to explore the D614G mutation. In the process, the virus did mutate more so, and we added to our project. But it is centered around that one, and that's the one you'll hear about first.
Yes. Hi, everyone. We did a little different presentation than using our tutorial, so I hope you guys don't mind. Our research was on primarily the D614G mutation in the spike protein. And we also looked into the United Kingdom mutant, which is the deletion of 69 and 70, and the South African mutant, which is the K417N and E484 mutations. And so the main mutation, D614G, is like the main thing that influences it is the ACE2. And that's because that binds with the protomer in the open conformation. And the reason that matters is because when it's in the open conformation, there is this, basically the spike protein is kind of this bulb, and when it's in the open conformation, the petals open and allow for binding. And when it's closed, that D614 mutation is significant because it has a hydrogen bond that breaks after the mutation. So you can see in the paper that inspired this project, it goes from a 2.7 angstroms bond length to an 8 angstroms, which is too big for a hydrogen bond. And that allows it to assume that open conformation much easier. And our research matched that. So you can see we got similar values when we studied it. So, oops.
So we also looked at the United Kingdom variant, and in this variant, we found the deletion of 69 and 70, those position of amino acids. And then we also found recently that it also included mutations in 484 and 501, which are also seen in the South African variant. The deletion of the amino acids at position 69 and 70 cause an allosteric shift in the conformation of the subunit, and this shift causes a blockage of the antibody interactions. And as you can see in our model, we have the deletion in salmon and the chain in blue. Okay, and then at position 501, this is seen in both the South African and the UK. And 501 is located in the receptor binding domain, but specifically the receptor binding motif, which plays an important role in structural stability in the receptor binding domain. And the exact relationship of why this has a higher affinity for the ACE receptors is still being researched fully, but it does cause a higher infectivity.
Now going to the South African, we see some of this mutations we just saw earlier that Trisha explained. We have the mutation 484 and 417 as well as 501. The 484 and 417 are in the receptor binding domain of the spike protein, and these mutations increase the affinity this protein has for the ACE receptor, and that allows it to have higher infectivity with this mutation. These are our resources we used, and ultimately, why do we care? Why are we doing this? Well, it's interesting, even just in the extent of our doing this project, the virus has already mutated so much so that we thought it was worthwhile to add on even more of these mutations. The D614G is now extremely common as an allele, which was once a mutation. We can talk about things like D614G in numbers and letters, but this allows us to see otherwise invisible changes and examine how they might interact with the proteins.
You guys have your model with you?
Yeah, we can show those. This is our active site model. Sorry, it's kind of hard to see. And Dr. Franzen made us this really cool like magnets. We can clip on the mutation, so you can see how it like changes in size from like, you know, an asparic acid, which is kind of big, to a little glycine. So I'm sorry, it's really hard to show because I can't see the camera while I show it. And then Trisha, do you want to show the other one?
Yeah, this is our other one. And as you can see, the protein is kind of like a flower, in which this was the receptor binding area that I was talking about. And I don't know if you can see them, but there's tiny little mutations in them. And the yellow is the, oh, geez, it's right there, is the 417 and 484. And this little green one right there is the 501. And the 484 and the 501 is a really big one that we're going to be looking into further, I think, in the future, just because the UK variant is becoming the dominant strain in the US. So thank you for listening to our presentation.
So could you hold that up again and show people the difference between the open and the closed conformations?
Yes, so this green is open, and as you can see, this part right here opens up towards the top, and then this blue and pink is closed, and it's closer to the bottom, so it folds in. That's what they were talking about whenever they talk about the petals. So these two would open up whenever it tries to bind to the ACE receptor.
I think you guys were very fortunate to find a structure that had both open and closed in the same structure to be able to show that. Dr. Schiffer?
Very nice presentation. You commented on how the aspartic acid to glycine mutation was a size change. I wonder if you could comment a little more on the different physical chemistry properties of some of these mutations, because they're really quite dramatic, even more, not just the size change, but other properties as well.
I forgot how to unmute for a second. Yeah, thank you for your question. If you don't mind, I'll share quick my JMOL so people can visualize like what this is. And so like, as you can see, aspartic acid is not only bigger, but it has like these polar groups on it, which are, you know, they interact with it as well with the hydrogen bonding. And that's, you know, those oxygens are what are binding. So if I click this, this shows like it goes from I know that was fast. It goes from an aspartic acid to this glycine, which is much smaller, and it doesn't have that oxygen to do the bond with this. And instead it has to form bonds with its side groups, which don't show in this, sadly. But so that really shortens the length, or lengthens the bond, and it goes to like, I think it's 8 angstroms, and that's just too big for hydrogen bonding. So that interaction is crucial. And I don't know if this answers your question, but we also noticed that because it shortened, it actually increased interactions with this one, with this 484 or 447.
Do you know another special property about glycine?
It's very non-polar, I believe.
And it's also very flexible. It's much more flexible than any other amino acid. So it gives more flexibility to the protein.
Okay, Feza?
Great presentation. I just had a quick question. So your team mentioned that the absence of that key hydrogen bond is what allowed it to assume that open conformation and therefore like increase infectivity. I'm just curious, are there any other key mutations in that spike protein, such as the one at the position of 614 that, or somewhere else, that have been shown to increase the infectiousness or transmittability of that, I guess?
Absolutely. Thank you for your question. Actually, that was one of the things that we were going to talk about a little bit further, but position 484 and 501 have been seen to cause allosteric shifts in the flexible loop. And this is because
the aspartic acid, as seen in 501, is non-charged and polar. However, the tyrosine is hydrophobic, and I believe there's also an aromatic ring on that, which is completely different from the amide that is seen in the aspartic or asparagine, sorry. And then 401, there is a shift in the charge of the amino acids. So it goes from a negative charge with the ketone and the oxygen on it to an amine in the lysine. And so this creates a positive charge. And they're still doing further research on that, but that's what's up to date.
Excellent. Thank you so much. And the next group is the MSOE team that looked at the spike protein, but you looked, instead of looking at mutations, you looked at an antibody binding to it.
Yes, that's correct. As part of our introduction to it, I have like a short, let's see, share. So can you guys see my screen? So we studied the cross-neutralization of SARS-CoV-2 by a monoclonal antibody that's also effective on the SARS-CoV-2 antibody on spike protein. So basically, this is the spike protein that we're studying, the spike protein of SARS-CoV-2. And on the top, you can see here, there are these glycans. They're oligosaccharides that are covalently linked to the surface of the protein. There are 3 of them, 1 on each trimer. And these serve as recognition points for the antibody. They are composed of several different sugars. There's mannose, fucose, beta mannose, and these are important markers that are also extremely conserved across all of the related coronaviruses. So they serve as important antibody targets. Here you can see the antibodies binding to the protein. They bind non-competitively and also serve as effector molecules for the immune system as they mediate antibody-mediated cytotoxicity. So they allow natural killer cells to actually bind to the virus and kill it. A boost to show the actual significant amino acid interactions, which is what we actually studied. We found out the using JMOL, the various significant amino acid interactions based on how close they were to each other. We have hydrophobic pockets in yellow, ionic bonds in green, and then we have some others. I will show you the JMOL exploration that kind of goes into a little more detail with them. So here you can see that we have a ionic bond between a glutamate and a lysine here. Then we have a yellow hydrophobic pocket. And then we have these purple struts here. Those are all dipole interactions. And the purposes of studying this was to kind of figure out why the antibody binds to the protein the way it does, and to help develop further treatments for the coronavirus, because the way the antibody binds is so conserved across the spectrum. We figured that if we know how it binds, it could be used as an effective treatment due to mutations that can arise and can make vaccines less effective. So having this as a backup treatment is always a good plan. Does anyone have any questions?
In your poster, you mentioned that you determined which residues were close to the antibody. How did you do that?
So we used a function in JMOL that uses the distance in between the residues as a point of comparison for how strongly they can actually interact. I believe the program deems anything less than 5 angstroms somewhat significant, and the closer they are, the more significant it shows. They have these red circles that grow bigger the closer they are together. And we use the complementary determining loops mentioned in our paper that are these areas of the spike protein and the lichen that actually the antibody recognizes. So by searching along those loops, we identified these significant interactions. You have a hydrophobic pocket here, some hydrogen bindings up here. There's one here between the backbone, and up here between it's a lysine. And these all serve to kind of help position the oligosaccharide and the spike protein to actually bind to the antibody. What was really surprising is that there's actually an ionic bond, which is a lot more stable than the others, because the rest are very dependent on hydrophobic pockets and weak hydrogen bonds and dipole-dipole interactions.
So Margaret, before we continue, there's a question on the chat, and I don't. Do you want to hear that now, or do you want to let?
Yes, go for it, because there's nobody.
Okay, well, Dr. Schiffer has a question too, but I'll go ahead and read the one on the chat first. Did you look at the N and O glycosylation profiles of the spike protein? And if so, could you possibly expand on that?
Can you repeat that?
Did you look at the N and O, so asparagine and O glycosylation and profiles of the spike protein? And if so, could you possibly expand on that, on the glycosylation profiles as I understand it?
I am not familiar with those terms. I cannot answer that question. I apologize.
Okay. Dr. Schiffer, and then Matthew.
A very nice presentation. I was curious, as you know, you've identified these different hydrophobic clusters and ionic clusters. When you look at different spikes from the different coronaviruses, or even as the mutations that are arising, how conserved are these sites? Would you expect this antibody to be able to neutralize across different forms of coronavirus?
Yes, the paper did say that this area is highly conserved across all sarbecoviruses. One reason for that is the glycans. Actually, in our reading, we found that often viruses use these glycans to kind of disrupt the surface of the spike protein that can help it evade the immune system. But in this case, that glycan is a key factor for actually recognizing and binding to it. So our understanding is that it's not a site of high variability and that it is highly conserved across all the coronaviruses that were mentioned in the paper. And that was experimentally verified by comparing the different crystal structures between them.
Okay, Matthew.
Hey, great presentation overall. So my question was, so you talked about the S309 binding competitively to the conserved, or non-competitively, I'm sorry, to the conserved glycan. And I was wondering, how exactly did you determine that?
It was a key point in the paper we read. It mentions that they did, I don't remember the exact test they did, but they did confirm that it does not compete with the ACE2 inhibitor. I cannot elaborate further on that. I believe it's because the CDR loops that it recognizes don't seem to really overlap with the areas that interact with the ACE2 receptor. So it's just the positioning of the antibody and its binding is what makes it non-competitive. It's not interfering with that, but it does effectively neutralize the virus, possibly due to a conformational change, or as I said before, it does work with the immune system to help natural killer cells bind to the virus and inactivate it.
Thank you.
Dr. Guzman.
I had two questions. One, I thought it was a very neat animation you started with. How did you do that? And then the second question is, you defined the different interactions in your JMOL exploration into great detail. So do you have a program that broke it down to dipole, et cetera?
To answer the first part, to make the animation, I use open source software called Blender. I use JMOL to isolate the initial structures, and then I export them as an X3D format, which is 3D printing. I imported that into Blender, and then I individually grouped all of the atoms and colored them. And then I used various procedural animation techniques and shape keys to kind of make them flexible and move, and assign those positions over a timeline to create the animation you see, as well as various procedural shaders to create the blood vessel and different lightings. The second part of the question is how we identified the interactions. What we basically did is, I should have a file somewhere in here that shows the actually interactions. I keep my files on OneDrive, so it takes a second to download. But anyhow, what we did is, for these pockets here, when we use a command in JMOL called contact, what it basically does is it takes a distance between the two, each atom of the various neighboring residues, and it measures the distance. And if that distance is small enough to constitute like a significant interaction, that the intermolecular forces would be strong enough to actually exert a non-negligible force on each other, it would show yellow and red circles of varying sizes, and that would lead to us deeming it significant. And what we did is we went through each CDR loop. We go to like this yellow one, so it'll be easier to show in JMO. So like we'd take this tryptophan and this proline, and they're very close together. And I don't remember the exact commands we used, but they would show like a circle there. And the bigger the circle, the more significant the reaction. So we took all the most significant interactions, and then we determined based on the positioning of the amino acids relative to each other, what those interactions would be. Because just because you have two hydrophobic amino acids next to each other doesn't necessarily mean it's a hydrophobic interaction. Like there is a glutamic acid in here that would typically form like a salt bridge or something, but it's actually forming like a weak dipole-dipole interaction between the backbone. So we just used the program to determine how close they were together, and then based on the characteristics of the neighboring atoms, what those interactions would be.
Okay, Dr. Schiffer, do you have another question? Or perhaps her hand was just kept raised out. Okay, thank you very much. The contact commands that you used, I think are listed in the JMO resources page on how. So if someone else is interested in playing with that with another group. Okay, our next group is NOVA, and they also looked at the spike protein and a different set of antibodies. So go ahead.
Okay, I'm gonna go ahead and share my screen. All right, so our team focused on how molecular models would help us predict a more effective antibody, more universal antibody against the SARS-CoV virus. And so we first started just studying the background of the spike protein and how it enters the cell through the ACE2 receptor protein. And then our next step was to understand how antibodies neutralize SARS-CoV and how it blocks the, attaches to the protein and blocks it from attaching to ACE2. And then when we were researching this, we came upon a Schiffer lab paper that studied the ADR antibody, which is known to bind to the SARS-CoV-1 spike protein, and compared that to the a new naturally produced antibody, MAB362, which they found to bind to both SARS-CoV-1 and SARS-CoV-2 spike proteins. And this right here is the MAB362 structure. So from comparing these, we observed a trend in the binding interface between the antibody and the spike protein that showed MAB362 amino acid compositions to be smaller and less polar, which created more space in the binding domain, indicating perhaps more space in this binding interface increased resistance to to the spike protein and various other variations of the structure. So in this picture, as you can see, on the left is ADR bound to SARS-CoV-1 spike protein, and in the middle is MAB362 bound to SARS-CoV-2 spike protein. And you can see clearly that the binding interface on the left structure is much more crowded than the one in the middle. And so using these trends, we proposed 4 additional amino acid changes to create a hypothetical antibody that would be more universally effective among the current and perhaps future variations of the virus. And this is NSU here, NSU1 that we made with the changes from MAB362. And this is a results table that is color-coded based off of the JMO models and highlights the 4 amino acid changes that we predicted or proposed that highlighted in pink, and it also shows the various changes from SARS-CoV-1 to SARS-CoV-2, as well as ADR to MAP362. And I will be handing it over to Lila to show our models.
Okay, hello, I'm Lila, and so I'm just going to be showing you guys the models. So basically 3 models were prepared to show the 3 different monoclonal antibodies, which were ADR362 and a hypothesized new antibody that binds to the SARS-CoV receptor binding domain of the spike protein. So this is our first model, model A, which is the ADR antibody with the, hold on, there you go, the SARS-CoV-1 spike protein. And then our second model is the model B, which is—
Lila, are you able to remove the background? Because I think it would be a lot easier to see the models.
Let me do that, 1 second. Okay, there we go. So this is model B, which is the 362 antibody with the SARS-CoV-2 spike protein. And then model C is a hypothetical new inhibitor, which we named NSU1. So here you can see that in the white sections, those are the hydrogen bonds, and then the yellow bars are struts that are meant to provide stability to the actual model. And then we also placed some magnets to allow the spike protein and the antibody to be separated, basically. And so to first design our antibody, we looked at a paper called the Antibody-Specific Model of Amino Acid Substitution for Immunological References from Alignments of Antibody by Mirsky in 2015. And we basically analyzed the different mutation trends and then modeled NSU1, which is this one, after the MAB362, based on the 4 following mutations. So we mutated aspartic acid to glycine at position 103, tryptophan mutated to leucine at position 104, glycine to serine at 170, and then arginine to valine at 211. And then in addition, 9 spike epitopes for model A and B were identified, and those are the ones that are colored in tan. So the tan colored ones. And so then at the end, we were just able to make something new that is predicted to be more resistant to future COVID-19 spike protein mutations, and overall could just be more universal. So we can take any questions now.
Dr. Schiffer?
Thanks for looking at our models. That's very cool. I wonder, so the model that we made is actually a model as well, and maybe your gap is picking up some things that aren't quite correct. We weren't able to get a crystal structure of that complex. So seeing that gap, I'm wondering, well, maybe something's not quite right. But I really like the fact that you decided to introduce mutations to try to make it fit better, and that's exactly the right thing to do. So thank you.
Dr. Martinez?
Hi, very nice presentation. Maybe you mentioned this and I missed it, but I was curious about how you were selecting mutations to test for potential resistance, etc.
So the 4 mutations we chose were based on the trends found that happened naturally from ADR to MAB362, which they were smaller and less polar to allow for more space so that future mutations it would be able to fit in better. So that's basically why we chose those 4 mutations.
Thank you.
So I was wondering along those lines, if you're making the pocket bigger, is it possible that you could make the opening so big that they wouldn't interact at all?
I think it's definitely possible, and I think that's why we started off with just 4 mutations. But yeah, I do think it's possible, and I think we should be careful about that.
And do you have any idea how you would go about testing to see if your new proposed antibody would work? Not saying be able to do it yourself, but if you were in a research lab, what might you do next?
We could try like testing up, actually creating the antibody, and then maybe releasing it only to SARS-CoV-1 patients in comparison to controls, control antibody treatments, and also the ADR antibody treatment, and then also release the our NSU1 antibody treatment to SARS-CoV-2 patients, and again in comparison to controls and to the MAB 362, and then test the efficiency.
Okay. I would encourage you, before you stick it in people, that you test it in cells and in tissue culture. Because what if you kill people to see if it works first?
Oh yeah, definitely. Sorry.
That's okay. You came up with a good answer, and it's a good logical answer. And just to say, I have been talking to some friends who didn't want to get vaccinated, and they were concerned that we used tissue culture cells to test whether the vaccines worked. And I said, well, it was unethical to test them first in humans without making sure it worked, because you should see if it works before you see if it works in humans. So that was great. Well, thank you so much. And our last team is the Campbell team that looked at the ORF8 protein.
Hello, everyone. Can you guys hear us?
Yes, you're good.
Okay, so we looked at the SARS-CoV-2 ORF8 accessory protein, and it's highly variable, and it's believed to have a role in immune suppression during infection. There's evidence that it inhibits the type 1 interferon pathway and downregulates MHC1 expression. So we designed 2 models of ORF8. One was analyzing the homodimer and the interactions between the monomers. And the other model analyzed how the homodimers can interact non-covalently to form an oligomer structure. And we also utilized theoretical docking software to generate a potential model for an interaction between ORF8 and the interleukin 17 receptor.
So let's take a closer look at the models. And this is the dimer right here. It's being held together by covalent and non-covalent interfaces, where we have hydrogen bonds, and then it has a giddy sequence, which is a tyrosine, isoleucine, aspartic acid, and isoleucine amino acids, which form non-covalent interactions to hold the dimer together. And these dimers can come together to form monomers, or oligomers, where we have the Yiddy sequence interacting and hydrogen bonds in covalent interfaces holding the oligomer together.
All right, and then to show you it a little bit clearer, I'm going to go ahead and share the screen so we can show you our exploration. Can you guys see that?
Yes.
All right. So while it's loading, we're going to show you guys the covalent interfaces involved in the dimer and the oligomer. All right, so first we're going to be fading it away, and then in the model, we'll be displaying our disulfide bond in yellow, our hydrogen bonds in pink, and then the salt bridge, which is the ionic bond in purple. These are, again, are the 2 identical polypeptides that form the homodimer. It should zoom in just a second. We colored the N terminus and the C terminus as seen. So that is the disulfide bond. And adding it back, that's how the homodimer would come in together to form the oligomer. And then for the dimer, I believe. And then to show the dimer, that is our model. Again, the hydrogen bonds are in pink. The unique disulfide bond is in yellow. The hydrophobic interactions are orange. And then the purple is the salt bridge. Do you guys have any questions?
Carol.
Hi, your presentation was really good. I just had a question about if there are other accessory proteins with similar functions as the one you chose, and if so, why was the ORF8 accessory protein chosen?
So there are other accessory proteins. The complete functions of all of them haven't necessarily been determined yet. We chose ORF8 because of the role it seemed to have in immune suppression, and yes, we wanted to look at the structural relationships that could be there.
Okay, I thought I saw another question. Dr. Schiffer.
A very nice presentation. I noticed that this ORF8 structure is almost entirely beta in your figures and your models. Do you know if it makes a similar fold to an antibody type fold, like a fibronectin type 3 fold, or is it not? Do you know what kind of fold it is?
The article, or the original article for this structure, did describe it as an immunoglobulin type fold.
It has some internal disulfide bonds.
Yes.
It's interesting. I wonder how it evades the immune system with maybe as a decoy antibody kind of response. It's really fascinating.
Dr. Martinez.
So there is a question in the chat from Dr. Melanie Van Streit, and she's asking, the Brazilian variant has a mutation in E92K at the dimer interface. Do you think this will impact dimerization?
We didn't look into that but that's a very good question. It's in between the back. We really can't say anything specifically to that, but that is a great question.
Dr. Schiffer, do you have your hand up for the question?
No, sorry, I just didn't take it down.
Okay, okay. Could this team tell us a little bit about, I think you used a file that wasn't published in the PDB. So how did you get that file? Tell us a little bit about what happened there.
We contacted the researchers that were involved in designing the original structure that was deposited into PDB, and we were fortunate enough that they contacted us and actually helped us to get the oligomer file in addition to having the dimer file, and they were very helpful.
Thank you. Dr. Guzman.
I was just going to say, they may want to point out why did we need the dimer file when there was already one published? And we did look at some things about another variant.
So we needed the other dimer file because there was a small problem with the file that was in PDB at the time. There was one amino acid that wasn't oriented in the right way. So where hydrogen bonding interaction was, it was a much longer distance away than it was. Since it's a homodimer, you can see there's two of each of those hydrogen bonds. So you can see when it's not the same on one side versus the other. So we were able to get a corrected structure with that reposition.
And then this is really a lot of fun because all of you worked on projects that were hot off the presses. And when things are hot off the press, sometimes they come out with some errors. And the PDB is a dynamic repository in that they can go back and correct errors, but you were looking at it before the errors were corrected in the PDB. So that's really cool. Dr. Guzman, do you have another question? Is your hand just still up?
Yeah, I meant to lower it, but I think Ryan was going to tell us about the other variant.
Yeah, so we actually were looking at another variant that had a mutation in our YIDI sequence, which is involved in constructing the oligomer. The mutation is where the tyrosine at the 73 location is converted to a cysteine. And from the articles that were reviewed, it looked like this could possibly lead to the formation of new disulfide bonds. But unfortunately, they didn't go into much elaboration on what they meant. They just were explaining that it would likely lead to a more transmissible strain.
Any other questions? Thank you very much. And if you can unshare your screen, that would be awesome. You guys, I just have to say, I'm really proud of all of you. This was a challenging thing. It's hard not to be in the middle of interacting with people with your model and a poster that you can point to the pictures, and even meeting together, figuring out how to meet and do the CREST project this year. I'm just so pleased with what all of you have done. And I hope someday we'll meet up somewhere that I can meet with you face to face and just get to know you guys better. But I also want to thank the faculty advisors, because I know this was a tough year for them, trying to juggle all the crazy differences in teaching, and yours with learning too, and still being able to help to develop the CREST teams and keep them going. Everyone has shown a great deal of resilience and flexibility. And I would have been happy if we'd had 2 teams this year. And the fact that we had 8 is a real credit to all of you. So at this point, I want to pass this over to our external evaluator. And for those of you who are not students, or for those of you who are students but are not yet 18 years old, can I confirm that you don't want anyone who's not 18? Is that correct?
Yes. Yeah. So I have to have 18 or older students.
Yes. And the rest of us have to be.
Project.
So Kim, I'm going to pass over to make you the host.
Okay. Excellent. Thank you.
And then I'm also going to stop. I don't see where to stop. Oh, I'm going to stop.